Alrighty, so we'll move into the next talk. And so Andrew Mackay from the Illinois Natural History Survey, and I'm really excited about this talk because we haven't really had any talks similar to this in the last few years. So it's going to be a new kind of talk to us about some invasive mosquitoes.
All right. Yeah, so I'm going to present some of the data that we collected from a recent study looking at some important population level traits of a relatively recent invader to Champaign County, the Asian tiger mosquito. So the Asian tiger mosquito has somewhat of a cosmopolitan distribution. It's found throughout temperate and tropical areas globally. In the U.S., it was first found here in Texas back in the mid-'80s. Within a couple of years, it popped up in Illinois, mostly confined to the southern part of the state. There were a few very small focal outbreaks in Cook County associated with tire treading facilities. But it really took a long time to really fill its ecological niche space in Illinois. And it wasn't until 2011 that it popped up in Champaign County. So we were really interested in looking at some of these important life history traits in a population really sort of at the extent of its range. So like all mosquitoes, it has this bifurcated life history where it undergoes development and growth in an aquatic system and then emerges as a terrestrial adult for reproduction and dispersal. And so the juveniles are mainly found in artificial container habitats, either discarded things like tires that fill up with rainwater, and a number of human-managed containers as well, like planter pots where people are adding water. So it really provides some plasticity to their seasonal phenology. They're not necessarily completely dependent on rainfall. And so the main significance of this mosquito in Illinois is as a nuisance pest. It's an extremely aggressive species. It's day active, and it prefers to feed on mammals. And it also has this high affinity for using these artificial containers, which really brings it in close proximity, both in time and space, with people. It is known to be a competent vector for a number of human pathogens. In Illinois, one example is lacrosse virus. We don't see a lot of cases, but it is known to be a competent vector. One of the real concerns is with some of these exotic human pathogens like dengue, Zika, chikungunya, which aren't endemic to Illinois, but we do have travelers coming from endemic areas bringing the virus in annually. And there's a concern that the introduction of these pathogens could allow local populations to at least support a very limited transmission of these human diseases that carry a really large public health burden globally. And then thirdly, these mosquitoes are notoriously very difficult to control. So they sustain a fairly high population density for an extended period during the season, and they utilize these very productive habitats that are widely dispersed through urban landscapes, both on private and public lands. So for vector control programs, public health programs, it's very difficult to target their larval habitats. So the two main aims of our study were to characterize these two very important population level traits, but important in terms of determining their capacity to transmit a pathogen. One is the human biting rate, the frequency they come in contact with people, as well as the population age structure, so their daily survival, their average age within the population. We wanted to look at this temporally across the season and also look how it's influenced spatially by its environment, and particularly looking at the influence of canopy density. So as I kind of alluded to, the reason these two are very important because they really determine the potential for disease transmission. Human biting rate determines the number or proportion of mosquitoes that are actually coming in contact with humans, possibly picking up or transmitting a virus. And the population age structure or daily survival determines the proportion that are actually going to live long enough that they can pick up a pathogen from an infected human and then pass that on to a susceptible individual. And this can involve a fairly long duration or age at which a mosquito is actually able to or at risk of transmitting a pathogen. So these traits are really influenced quite greatly by the environment. And one major environmental factor is terrestrial vegetation. And it has a lot of both direct and indirect effects at different points in the life history. With the juvenile life stages, one of the main influences of terrestrial vegetation is providing this substrate within the aquatic environment. Decomposing leaf litter really drives these food webs, creates microbial blooms that the larvae feed on, and it really determines the sort of carrying capacity of these environments, how many adult mosquitoes are going to actually develop in a container. It can also have very profound effects on the fitness of the adults. So nutritional limitations or phytocompounds that are released by these leaf substrates can, say, impact the longevity of these adult mosquitoes. And then in the adult life stage, plant or terrestrial vegetation is very important for providing nutrition. So both the males and the female adult mosquitoes really rely on things like nectar for obtaining energy for flight. So the distribution and quality of floral resources in their environment might have a very large impact on their daily survival. And it also structurally provides very important resting sites that also enhance their daily survival. So we set up a study this past summer in 2018 in Urbana-Champaign to look across a residential landscape that varied in canopy structure to look at how this human biting rate as well as the population age structure and Aedes albopictus populations varied. So we used U.S. Census blocks as our spatial unit for defining the environment, canopy density. And this is based on their size. It's an appropriate size for defining the area of environment that's likely to have population level effects because it approximates the average flight range of these mosquitoes. And it's also bound by these relatively inhospitable open road networks, which provide some impediment to dispersal. So we used a fine scale land cover map, classified primarily residential census blocks across both communities, defined moderate canopy class as well as a high canopy class, selected for residential census blocks for each class, and then identified an appropriate home a single home within each of these census blocks to actually sample mosquito populations. And so at each of these 8 homes, the 4 medium canopy, 4 high canopy, we sampled weekly across the season. and we sampled host-seeking mosquitoes by 2 methods. I'm going to talk mostly about results from this first one, the human landing collection. This is the one that provides the most direct measure of human biting rate, and it involves having a person in a seated position in a nice, very sheltered habitat where they expose the one lower leg and then just use a small handheld aspirator and collect all of the mosquitoes that land on their exposed skin surface. And we collect them at 5-minute intervals over a total 30-minute period. And by collecting them at intervals, we get a more direct measure of say, the effect of exposure time on the number of bites. Because we're interested in looking at population age structure, we also took these live specimens that were collected during the landing collection, took them back to the lab and measured their residual or post-capture longevity under optimal conditions. And although this is still an ongoing project, we're intending to use this to apply something called the captive cohort method, which is a demographic method for inferring something from this residual captive longevity to tell us something about the actual population age structure, the age structure of the population that was sampled. We also used a BG sentinel trap to sample mosquitoes. This uses a artificial human lure to mimic a human, and the samples collected by this device will then be used to also apply another aging method based on the morphology of the female ovaries. So we'll have 2 independent measures of age in our samples. So this is just a really sort of simple schematic of the captive cohort method. As I said, this is still a pending analysis. Today I'm going just to present some of this raw post-capture longevity data, and it can be roughly considered sort of the inverse of what we expect the mean age of the population that was sampled. And this just shows the main assumption of the captive cohort method is that if you take a sample and measure the length of time individuals in that sample live and take the mean age at survival, this should have an inverse and proportional relationship to the average age of the individuals in the population. So if you have a very long survival of the sample in captivity, that should reflect a very young population, and the inverse is true where you have females dying very quickly in captivity. That would indicate a very old population. So I'm just going to present results first sort of on the temporal dynamics of human landing rate and captive longevity, and how that relates to the season, and then talk a little bit about spatial relationships in association to the environment, canopy density and distribution. So with both methods, we found Aedes albopictus was the main or the most common species associated with either a live human or a artificial human lure, indicating this is probably the most common species people are coming in contact with in the home environment. And this figure just shows the seasonal distribution of human landing rates. So these represent the average number of females collected per 30-minute landing collection, averaged across all 8 sites across the entire season. You can see there's a lot of seasonal variability. There's a very considerable variability among sites. But generally, we had fairly high landing rates through much of the season, well over 10 females per landing collection, which is considered a pretty high human biting burden. So another way to look at this, so this is the same data from the previous slide. aggregated across all sites, across all dates, but segregated by the individual 5-minute intervals for each landing collection. So this represents the cumulative number of bites that a person might be expected to receive, depending on how long they are out and exposed in the environment. So the main thing to gather from this figure is that even very short exposures can result in a very large number of bites, indicating that even these very brief outdoor activities really warrant taking precautions like using repellents and such. And so this, how this compares with other areas throughout the globe where Aedes albopictus has a much longer history of establishment. We found, so these are all of these individual colored points represent previously published values for human landing collections that were performed in areas where Aedes albopictus has been long established, and there's either been a history of disease transmission, or the authors have described this as a location where Aedes albopictus has an unusually high abundance. We found that our observed results really fell within that range, which indicates that the density of Aedes albopictus in Champaign County is quite high, despite being a relatively recent immigrant to the county. And so then moving to captive longevity. So this can be considered the inverse of the average population age. So where you have a brief or a much shorter captive longevity, this indicates probably an older population, and higher captive longevity would indicate a younger population. So what we saw is similar to biting rate, there was a really pronounced oscillations throughout the season, but not nearly as much spatial variability. So there's a lot less variability among sites. However, this is just raw captive longevity data. At this point, it's not clear what is driving a lot of these dynamics, whether it's either, say, changes in recruitment, so you have a greater number of very young females entering the population and increase in the captive longevity, or whether these are actually changes in the maximum longevity. So there are periods where the maximum lifespan is changing in the population. But based on some of the comparisons with weather data, they seem to more support the sort of former idea that a lot of this is driven by just changes in the rate of production. So this just shows scatter plots for average captive longevity for each of the 18 weekly sampling periods by either the total sum rain, approximately 2 to 3 weeks prior to sampling, and temperature for a shorter lag, 1 to 12 days before. And so these roughly correspond with when you would expect them to have an influence on the youngest females that would be emergent or being collected in our landing collections. And so what these results suggest is that higher rainfall 2 to 3 weeks prior to each of our, or prior to a landing collection, followed by a higher mean daily temperatures, are increasing the rate of recruitment, and that's what's driving a higher captive longevity value. And so the other main aim of this study was to look at the environmental effects of canopy structure. So one measure was this overall density at the scale of a neighborhood, a US Census block. And unfortunately what we found is there was really no or very limited effect or relationship to either of our two measures. We didn't see any differences between the high and moderate canopy in landing rate and really minimum differences with captive longevity. We did see some interesting associations, at least with more fine-scale differences in the distribution of canopy vegetation on human landing rates. So this is just one example here where we had a very positive relationship between the size of the largest canopy patch overlapping the home where the landing collection was done, and the number of females collected in the landing collection. Similarly, with captive longevity, we found that the number of patches of canopy at the neighborhood scale, so the surrounding census block, had a negative relationship to captive longevity, and suggesting the inverse with mean population age, so associated with an older population. We also found a relationship with the landscape shape index, so a more complex canopy environment with a lot more edge habitat was associated with a lower captive longevity and presumably an older population age. So before I summarize these, I really have to put sort of the caveat, this is an ongoing study, and a lot of these analyses and observations and interpretations are somewhat preliminary. However, we feel very confident that certainly the sorts of densities we're seeing of human-biting Aedes albopictus in Champaign are consistent with a very high, very well-established population of the Asian tiger mosquito. So it's been very successful in Champaign County, and is now a really important pest and may represent a public health threat in terms of disease transmission. We saw a substantial variability across the season in both of our measures, captive longevity and human landing rate. The latter does appear to be associated with antecedent weather conditions, suggesting that it may be driven by recruitment. We do see some evidence, though, that some of the seasonal differences in mean captive longevity are associated with changes of longevity, which would have much bigger implications for disease risk if you have changes in the maximum lifespan of mosquitoes. And so some of this will be, I think, a lot clearer when we start looking at things like general measures of adult fitness. So one of the things we're looking at is mean body size. And if that is also associated with this increase in captive longevity, that may indicate that changes in maximum longevity are also driving these changes in captive longevity. So we didn't see any differences in or any influence of overall canopy density on either measure, but we are seeing some of these more fine-scale effects of canopy structure, indicating that they may at least provide some value for, say, developing predictive or risk models for both Aedes albopictus nuisance issues as well as disease risk. And so, as I mentioned, we still have a number of these analyses going. This is still an early part of the project, but hopefully these will provide some useful information in terms of really describing the risk that this invasive species poses to Champaign County. So I'd just like to thank some of the individuals that really helped us with this study, and of course the people that allowed us to sample mosquitoes, and acknowledge our funding, which is through the state of Illinois. Thanks. Oh, yeah.
Can the viruses that infect the mosquitoes go from one generation to the next?
Oh, you mean pass from the female to the egg?
Yeah.
There's some evidence for, at least in the case of some of these pathogens, that there's a very limited amount. It's probably not playing a large role in, say, determining the severity of outbreaks, but it might be an important mechanism for, say, survival of the virus. So in the case of the endemic one that I mentioned, lacrosse virus, there definitely is evidence in other mosquitoes. I'm not certain with Aedes albopictus, but so it really depends on the pathogen. Yeah.
I'm wondering if you've looked into whether or not the native mosquito predators will eat this mosquito too. If they're not eating it, would that be part of why they're growing so fast?
In these artificial container systems, there certainly are insect taxa that play a role in suppressing mosquito populations, but they tend to be have less influence on population dynamics. So there are a lot of interactions with other container mosquitoes that do that may play a role in determining the success of Aedes albopictus. It does tend to be a superior competitor, which may help it sort of expand its niche range in here. But I'm not aware of too many mosquito predators that really have a great effect in these sorts of habitats.
So by living out in the country and having bats, that would be a protection against these?
Not so much for this one. This is more of a day-active mosquito, so it probably wouldn't have a large impact on the adult population.
I'm interested in interactions with fogging, with urban fogging, and the fact that these are daytime mosquitoes. A, would fogging affect these populations, and B, would an evening fogging, which normally it's done at sundown, would that affect these populations, or could something from the fogging be affecting those ups and downs in your populations?
They're probably less susceptible. They, as I say, they wouldn't be active, so they'd probably be resting in the vegetation. I had recently seen there was some work that was done, I think, in New Jersey, where they did see an impact on Aedes albopictus populations from the sort of traditional evening fogging. And I think it was assumed that, because most of this was ground fogging, so that it was able to reach the resting places. Yes.
A couple of years ago, I started seeing a product in the big box stores that says you can put it in your bird baths and it will kill mosquitoes and it won't hurt anything else. Do you know anything about the effectiveness of that and if it really is safe for other creatures?
There are a number of products. The most common one is a microbial insecticide, BTI. And there are a couple of commercial products, and they have been shown to be effective, and they are quite safe in terms of vertebrate toxicity.
One more?
Oh, sure.
Going back to the life cycle, could you recap quickly what happens during days 5 to 15?
Oh, and that was the figure where I was showing the relationship to here? Was that? Or?
It's very early.
On its life cycle.
Oh, the life cycle.
Forward, one more, I think.
I'm sorry.
There you go.
Oh, right. So, these are just rough values. And this is describing what's called the extrinsic incubation period. So this is the period from when a mosquito acquires a pathogen, when it takes it up in a blood meal, to the point where it develops within the midgut, escapes, travels to the salivary glands, and gets in the saliva. So it's highly variable depending on temperature, the pathogen. And so I just gave this as sort of an indication that there is a long lag time until they're actually able to pass the pathogen on.
How long do they live?
It really depends, and that's something we're trying to determine. Average ages might probably the mean age is relatively short, and it's a very tail. It has that very uneven distribution, non-normal distribution. Usually, people speak in terms of maybe 1 or 2 weeks is the average age.
What do you mean by recruitment?
Just that you're having more births coming into the population, so very young individuals coming into the population through mosquito production.
How do they overwinter?
This species overwinters through a diaplasing egg, so they lay the eggs on the sides of the surface, and it's very desiccation resistant. It has some cold resistance. So that's why they're able to be spread through the used hire trade, as they have these very resistant eggs.
Thank you very much.